now we move to France. Dr. Karim Adjou from École Nationale Vétérinaire d'Alfort. So a paper on Cryptosporidium. Okay, we'll get you up now. Yeah. Okay.
The laser is this one.
Okay. Thank you, Michael. Good morning, everybody. So my presentation is entitled Efficacy of Chitosan, a natural polysaccharide against Cryptosporidium parvum infection in vitro, in vivo, in small ruminants. As you know, the cryptosporidiosis is a zoonotic protozoan disease which has been reported in large number of animals, such as young ruminants. The Cryptosporidium parvum is the main agent of the neonatal diarrhea in young ruminants, especially in calves, lambs, and goat kids. And this cryptosporidiosis in humans is a major health problem in the 80s and in the 90s because this disease affected immunocompetent and especially depressed persons, such as HIV-infected persons without access to antiviral therapy. And recently, it has been reported many cases of children infected in open farms. So, as you can see on this slide, the clinical course of the disease is acute diarrhea in small ruminants, here goats and calves. It affects these animals between the age of 1 to 3 weeks old. The conditions of the occurrence of the disease are still not clear. Environmental factors seems to play an important role. So actually, no drugs are fully effective in the treatment of this disease in man as well as in animals, and no vaccine is available. Therefore, the research for new compounds is very important. Many cell models have been developed in the world to cultivate Cryptosporidium species. And you can see here this table with the most important cell lines. Two of them are very interesting for us. The first one is human ileocecal adenocarcinoma cells, named HCT8, and the second one is human colonic adenocarcinoma cells, called or named CACO2. So these two cell models were selected in this study. What are now the aims of the present work? First, we wanted to set up in our lab in vitro basic culture methods able to support the multiplication of Cryptosporidium parvum. Second, we wanted to test the toxicity of the drugs, especially parvumomycin, chitosan NAG, chitosan mixed, in our models. And third, we wanted to evaluate in vitro efficacy of these drugs on the multiplication and intracellular multiplication of the parasites. And fourth, we wanted to test in vivo the efficacy of chitosans in goat neonates artificially infected with Cryptosporidium parvum per oocytes per oral route. So this is the materials and methods. As previously I mentioned, CACOS2 and HCT8 cells were selected in this study and infected with a referenced strain of Cryptosporidium parvum named strain Iowa, produced by waterborne laboratories in the United States of America. And the method used in this study is the immunofluorescence technique in order to identify and to quantify the intracellular parasites in our cell models using a polyclonal specific antibody SporoGlow. This scheme is a representative of the slide we used in the enumeration of the parasites. As you can see here, we count 6 columns with 50 fields randomly chosen on the whole the surface of the slide. Now, a few words about the drugs. We have parvumycin. As you know, this is an antimicrobial drug well known in human medicine and in veterinary medicine to treat cryptosporidiosis. This is our reference molecule. Chitosans are a natural sugar compound that has found to be active against a variety of diseases, including antimicrobial diseases, antitumoral, and antifungals. Chitosan is a natural product, non-toxic, biocompatible, biodegradable, and attractive candidate for drug screening. In this study, we selected 2 products. The first one is Chitosan NAG, which is, we can say, the purified molecule. And the second one is Chitosan mix, which is the mixture of Chitosan NAG and other polysaccharide. These 2 molecules were supplied by Le Safer Feed Additives company in France. And this is the preliminary studies of the cytotoxicity. As you can see here, this experiment was done in order to determine the optimal doses for the 3 drugs in these cell models. The tested cells are 50, 100, 150, 200, 500, 550, and 1,000 microgram per ml. And as shown on this slide, the optimal dose obtained in RCT8 cells are for parvumycin 500 microgram per ml, Chitosan NAG 750, and Chitosan mix 1,000. For the second model, CACO2, we have 150 for parvumycin, 750 for Chitosan NAG, and 1,000 for Chitosan mix. So we decided for our studies to use the dose of 500 microgram per milliliter. So this is the first results of the pre-incubation experiment. What it means, we took the drug, and this drug was mixed with oocysts of Cryptosporidium parvum ioa during 24 hours. And after that, we infected with this mixture the cells, the RCT8 cells, and after the CACO2 cells. As shown on this slide, you can see here that these 3 molecules are able to inhibit the multiplication of the parasite in vitro, especially in RCT8 cells, in comparison with infected, non-treated cells. And the reduction are very impressive. As you can see here, more of them are than 90%, a reduction of 90%. The same results were obtained in CACO2 models, as you can see here, and the reductions were more than 97%. So these results show clearly that this, in the pre-incubation study, these drugs inhibit the multiplication of the parasites. After that, we tried to study the effects of the drugs on C. parvum, but after treatment, in the situation of treatment. So the cells were infected, and 48 hours, we come with the drugs, and we put the drugs into the cells. And we looked to the results 2 days after. And as you can see here, these 3 drugs were effective in delaying the multiplication of the parasite in comparison with control. And the best result was obtained with Chitosan mixed. The same results were obtained in CACO2 cells model, as you can see here. And again, the best results were obtained with Chitosan mixed, with a reduction of 68%. So this in vitro results encourage, these encouraging results allow us to test these molecules in Vivo in a small ruminants model. So we selected 16 goats, neonates goats kids, and we divided these animals in 2 groups. Here, 8 animals were infected and non-treated, so they are our controls. And the other groups is a treated group with OptiSafe, which is a product which contains at least 18% of chitosans in his composition. And these animals were infected orally by Cryptosporidium parvum. And the treatment was at the dose of 12 grams per day during 1 week. And the aims of this study were first to evaluate and to record the daily diarrhea scores, second to monitor the fecal oocyst excretion each 2 days, and to look to the weight gains and compare the two groups. So as you can see here, the effects of OptiSafe on C. parvum diarrhea. Here in blue, you have the control. So in the control group, diarrhea started at 4 days post-infection with a maximum at 9 days and diminished since the 9 days post-infection. And the mean score is 3.3. For the treated group in red one, you have here the start at 5 days and a maximum at 9 days post-infection and a very important diminution. As you say, the décalage between the two curves. So this result shows that this treatment have an effect on the score diarrhea. Looking to the diarrhea, if it's solid feces, liquid feces, very liquid feces, or bloody feces. And now, this is the results of the effect of OptiSafe on fecal oocyst shedding. And as you can see on this slide, in the control, you have oocyst excretion started at 6 days, and after we have the peak at 8 days post-infection, and we have a decrease, a significant decrease, maybe due to the immunity of the animals. In the treated groups, you have the same, but you see the décalage between the two curves. And so this is the first result showing that this OptiSafe product has an effect on oocyst shedding. So in conclusion, I can say that first, these HCT8 and CACO cell models are good models for therapeutic screening. Second, that the two models, in these two models, pre-incubation treatment was efficient in inhibiting the multiplication of the parasite. Third, in the pre-incubation test, chitosans were more effective than parvumycin. Fourth, our studies provide the evidence for the first time for the inhibitory activities of chitosan against C. parvum. And finally, our findings showed the efficacy of OptiSafe in reducing the parasitoid shedding and diarrhea in goat sheep, in goat kids, cryptosporidiosis. I would like to thank all the collaborators. Thank you very much for your attention.
Merci, Kate. Okay, so interesting paper. Open now for questions. And again, if you could just state where you're from and make sure you have the mic. Can we get a mic down to Stuart?
Hi, very interesting. You're, it looks like you're going to need quite high doses in vivo. Is that right? Are the doses you need in vivo achievable?
Yes, because the molecule is not very toxic, and for human too. Because
what's its half-life?
About OptiSafe or chitosans?
Chitosans, yeah.
Chitosans, it's a sugar. It's only polysaccharides. Yeah. So we take it, for example, in humans for diet, like parts of oral roll at high doses. So it's not toxic.
No, I'm sure it isn't. Thank you.
Okay. Another question here.
Hello, thank you for the talk. My name is Sabine from Austria. I wonder how were the animals fed? Did they drink from their mothers during the trial?
No, we used they sucked the biberon.
Biberon, oui, je comprends. But it was natural milk or milk powder?
No, milk powder. It's not natural milk.
Okay, thank you very much. Okay.
Welcome.
Okay, we're going to keep moving on. We're going to move now to the Italian Alps. And an interesting, thank you very much again.
Thank you, Monica.
Thank you very much.